Hi everyone, we are going to start Unit 5 today with Chapter 40, which is the Introduction to Animal Structure and Function. We'll be going over a variety of topics with the focus being on the 4 different types of tissues. Alright, before we jump into Chapter 40, I wanted to take you back to the first day of class where we covered the levels of biological organization in an animal or in organisms. So at our very smallest unit, we have atoms. So an example of an atom would be carbon. A molecule is 2 or more atoms that are linked by covalent bonds. So one of the most important molecules in biology is water, which is joined together by polar covalent bonds within the hydrogen and the oxygen atoms. Then larger, we have macromolecules, which are large biological molecules or polymers that are made of repeating monomer subunits. We have those 4 classes of macromolecules that we learned about in Unit 1: carbohydrates, lipids, proteins, and nucleic acids. And then we have our cell structure, which includes things like ribosomes, and in eukaryotic cells, we have organelles, or the nucleus. Maybe we have things like flagella or pili, or cytoskeleton fibers. We learned a lot about cell structures in Chapter 6. Right? And then finally we have the cell. And the cell is a membrane-bound unit of life. And we learned about the difference between prokaryotic cells and eukaryotic cells. And we learned about the difference between cells and viruses. Within Units 2 and 3, we learned about cellular processes, which included metabolism and genetics. And then in Unit 4, we jump to the diversity of all animals. And now here we are in Unit 5, and we're going to compare the tissues, organs, and body systems. And we'll do this using mammals or humans as our example. So I am going to shift a little bit from our book. Our book takes a comparative approach amongst all animals, ending with mammals. What we're going to do with all of our body systems is we're going to start with mammals, and then we're going to compare to other animals that we learned about in Unit 4. All right, so one of the key take-home messages from the entire semester is that each of these levels, composition, what something is made of, structure, what something looks like, and function, what something does, are all interrelated. So we can use water as a good example of that. The composition of water is the electronegative oxygen atom bonded to the non-electronegative hydrogen atoms. Because of the composition, the structure results in a partially positive end and a partially negative end, which we call a polar molecule. So the composition is related to the structure. And then the functions of water include the 4 major characteristics for why water is important to life. We learned that in Chapter 3 within the first unit. It was our second unit of the year. And because of the ability to form hydrogen bonds with other molecules, those weak bonds between a partially positive hydrogen atom on a water molecule and a partially negative charge on another molecule, because of this ability to form hydrogen bonding, water is essential to life. We saw examples of how composition, structure, and function are interrelated at the cell structure level. So for example, we learned about phospholipid membranes being a permeability barrier. And here's a brief picture of that phospholipid membrane. We have our hydrophilic region and the ionic tails. And we have the hydrophobic region in the fatty acid or hydrocarbon end tails. And because of this amphipathic structure, it allows for the bilayer to form with the hydrophobic tails on the inside and the hydrophilic ends on the outside. And this helps with the function of a membrane, which is that it is a permeability barrier to the cell. These hydrophobic tails keep hydrophilic stuff from passing through. And so as we progress to the tissue level and the body system level, we'll once again see that composition, structure, and function are interrelated. All right, so today's lecture will focus mainly on tissues. So let's review when we first learned about tissues. This was in Unit 4. We learned that the definition of an animal was that it is a multicellular, heterotrophic eukaryote with tissues that develop from embryonic layers. A lot of definitions, but we broke down what each of those mean. So embryonic development is something that is common to all animals, and tissues develop from those early embryonic layers, the endoderm, mesoderm, and ectoderm. All right, and so the endoderm is the innermost tissue lining. If we look at the gastrula stage, the endoderm would be this one right there. This is going to develop into the lining of the gastrointestinal tract, like the stomach lining, the small intestine lining, and some of the organs of the digestive tract, like the pancreas or the liver. The mesoderm tissue is the tissue that forms within the blastocele area. So this would be the mesoderm tissue right here that lines body cavities if you're a coelomate. And mesoderm tissue will develop into some of the internal organs like the muscles, skeleton, reproductive organs, kidneys, heart, lungs, etc. And then we have the ectoderm, which is the outermost layer. The ectoderm germ layer will later develop into the tissues of the skin or the outer layering, like the scales of reptiles, the central nervous system, and some of the sensory organs in the anterior portion of the heads of animals. All right. So in Unit 4, we learned that parazoa don't have true tissues, whereas eumetazoa is a clade consisting of all animals with true tissues, which means that these specialized cells group together and they're separated by membranous layers. Sponges are the only phylum, the phylum Porifera, the only phylum that has specialized cells, but as they are not separated by membranous layers, they are not considered true tissues. Diploblasts are animals that have tissues derived from two germ layers, whereas triploblasts have that mesoderm or third tissue germ layer that develops into more tissues. So remember that all triploblasts are bilateral. Our bilateral triploblasts can include acoelomates like flatworms, pseudocoelomates like the roundworms and rotifers, and many acoelomates and pseudocoelomates won't have circulatory systems or respiratory systems. And then we have our coelomates, which will. So coelomates will have the most internal organization. All right. One thing on your study guide, I want to point this out. Here is the PDF of your Unit 5 introduction. I have replaced what was in your course pack with a few, I've removed one of the chapters and I streamlined the study guide and took out some stuff that wasn't necessary. So this study guide includes a list of comprehensive topics, and then for Unit 5, I have some major big picture questions or concepts from each of the lectures. So from the lectures, the first concept is the importance of surface area to volume ratio among the different levels of organization. So we're going to talk about that in terms of tissues, but then relate that to what we've learned about organelles and cells. And so one thing we've learned before was that thin, flat animals don't require a circulatory or respiratory system. And this is because they have a high surface area to volume ratio. So remember that the surface area of an object is going to be in units that are squared. So let's say this is in millimeters. The surface area would be expressed in millimeters squared. Volume is expressed as millimeters cubed. And so if a cell gets bigger, let's compare this little bacterium to a larger yeast cell. The surface area and the volume are both going to increase in size as we get larger. So when we get larger, both surface area and volume increase, but it's not just the surface area that matters, it's the surface area to volume ratio, because diffusion has to occur through the interface and then all throughout the volume as well. So surface area will increase in square units, but volume will increase in cubic units. So in general, when we get larger, the surface area to volume ratio goes down. And that's because the volume is going to increase more than the surface area. So as life evolved, surface area to volume ratio goes down, and as animals are multicellular and more complex, they need ways to maximize this surface area, to limit the decrease in that surface area to volume ratio. So ways to maximize surface area include a lot of folding or protrusions like you see on this amoeba cell. Very angry kitty. Another way is by being very thin and flat. Right, so a cell that looks like this will have a larger surface area to volume ratio than a cell that looks like that. So our animals that are thin, flat, and exposed to water on all sides will often not need a circulatory or a respiratory system because diffusion occurs at a higher rate if the surface area is higher. And at the tissue level, we'll see certain types of tissues that are involved in gas exchange being very thin and flat. Another place that we've seen the surface area to volume ratio before is in the mitochondria. So the mitochondrion is an organelle that's involved in cellular respiration, and that inner membrane or cristae of the mitochondria has a lot of folding in it. And this folding allows for the attachment of more ATP synthase enzymes to really maximize the amount of ATP that is made. So an increased surface area allows for better diffusion and more attachment sites for important proteins.